denaturation and renaturation. Now, as we know that DNA changes their form, so how they will change? First they will break and then they will again join together, okay, by changing their form. So what happens? The hydrogen bond between nitrogenous bases of two strands of DNA, what happens? At a high temperature they break. Okay, so this breaking process is known as denaturation or melting. So what I told, the hydrogen bond present in nitrogenous bases melt at high temperature, or what we can say, break their bonding at high temperature. Now, this temperature range is there 82 to 90 degrees centigrade. So it is what? Melting. Tm means melting temperature, 82 to 90 degree centigrade. Okay. And what happens? These two strands of separated DNA happens. Okay. And these two strands separate from each other, and this process is known as denaturation or melting. Okay, where it will be having when temperature will be very high. Okay, now again when temperature goes down at 65 degrees centigrade, so what happens? These two hydrogen bonds which separate again join together, and this process is known as renaturation or annealing. Okay, so when temperature goes down, okay, when temperature goes down, at what temperature that is having goes down? At 65 degrees centigrade. So at 65 degrees centigrade, the hydrogen bond between nitrogen base nitrogenous bases attach to each other and form again two strand of DNA, two strand of DNA, and this process is known as renaturation or annealing. Okay, these terms will come. Okay, these terms will come in the lecture. Okay, so we have to understand that what do you mean by denaturation, what do you mean by renaturation. So denaturation occurs at which temperature? 82 to 90 degrees centigrade when temperature will come, and a renaturation occurs at which temperature? At 65 degrees centigrade when temperature will goes down, then they again join together, appearing that two strands of DNA, and that is known as renaturation or annealing. Okay, now next term is palindromic DNA. What do you mean by palindromic DNA? palindromic DNA. Have you seen one word just for one example? RADAR. It is radar. If we are going from this side or we are going from this side, both are reading same. So forward and backward, if both having same meaning, that is known as a palindrome. What I told? Forward and backward giving same sense. This is known as what? Palindrome. Now, since we are discussing about palindromic DNA, so what I told, one is GAATTC. GAATTC. Similarly, GAATTC. This is 5 prime to 3 prime. This is 5 prime to 3 prime. Okay, because they are having what? Anti-parallel in nature. DNA is when double stranded is forming. So how they are forming? DNA double strand is anti-parallel in nature. What we are seeing here, if we are going from 5 prime to 3 prime, same sequence GAATTC. If we are same 5 prime to 3 prime, so GAATTC, and we are seeing that this cytosine combined with guanine, thymine combined with adenine, adenine combined with thymine, adenine combined with thymine, adenine combined with thymine, guanine combined with cytosine. So in this way, if sequence of DNA are coming, that is known as palindromic sequence. So what I told, sequence of nucleotide is same but opposite in two strands. Sequence of polynucleotide is same but opposite in two strands of DNA is called what they are known as palindromic DNA. So if example are coming, so what are the examples are there? Very examples are there. But here we have seen GAATTC 5 prime to 3 prime, same opposite GAATTC. So that's why I told sequence of polynucleotide is same but opposite in two strands of DNA, and that is known as palindromic DNA. Okay. Now these sequences are recognized by restriction endonuclease and used in genetic engineering. Why this palindromic DNA is helpful, is beneficial? So what I told, these sequences are recognized by these sequences are recognized by which enzyme? Restriction endonuclease. This enzyme name you have to remember. Restriction endonuclease. Okay, and this palindromic DNA used in where? Are used in genetic engineering and used in genetic engineering. Very important. Why this palindromic DNA is being recognized by, this is recognized by restriction endonuclease, and why? Because it is used in genetic engineering. Nowadays many products are being formed during what? analysis or scientific research in genetic engineering. So this is what palindromic DNA. Now we will see that what are the difference between prokaryotic DNA and eukaryotic DNA. Okay, and that we'll see in the next class, and more we will go ahead. Okay, thank you. Please revise this one very carefully because today class is very tough. Okay, so be emphasis on this, revise many times. If any problem will be there, you just ask me. Okay, thank you.